हेलो फ्रेंड, फिफ्थ नंबर का हमारा चैप्टर है मॉलिकुलर बेसिस ऑफ इनहेरिटेंस। 5.1 द डीएनए, 5.2 द सर्च फॉर जेनेटिक मटेरियल, 5.3 आरएनए वर्ल्ड, 5.4 रेप्लिकेशन, 5.5 ट्रांसक्रिप्शन, 5.6 जेनेटिक कोड, 5.7 ट्रांसलेशन, 5.8 रेगुलेशन ऑफ जीन एक्सप्रेशन, 5.9 ह्यूमन जीनोम प्रोजेक्ट, 5.10 डीएनए फिंगरप्रिंटिंग. In the previous chapter, you have learned the inheritance patterns and the genetic basis of such patterns at the time of Mendel, the nature of those factors regulating the pattern of inheritance. Factors regulating the pattern of inheritance was not clear over the next 100 years. The nature of the putative genetic material was investigated, culminating in the realization that DNA deoxyribonucleic acid is the genetic material, at least for the majority of organisms. In class 11, you have learned that nucleic acids are polymers of nucleotides. The deoxyribonucleic acid, DNA, and ribonucleic acid, RNA, are the two types of nucleic acids found in living systems. The DNA acts as the genetic material in most of the organisms. RNA, through it, also acts as a genetic material, in some viruses mostly functions as a messenger. RNA has additional roles as well. It functions as an adapter, structural, and in some cases as a catalytic molecule. In class 11, you have learned that you have already learned the structure of nucleotides and the way there's monomer units are linked to form nucleic acid polymers. In this chapter, you are going to discuss the structure of DNA as its replication, the process of making RNA from DNA transcription, the genetic code that determines the sequences of amino acids in proteins, the process of protein synthesis, translation, and elementary the basis of their regulation, the determination of complete nucleotide sequence of human genome during last decade has set in a new era of genomics. In the last section, the essentials of human genome sequencing and its consequences will also be discussed. Let us begin our discussion by first understanding the structure of the most interesting molecule in the living systems, that is the DNA. Is subsequent sections we will understand why it is the most abundant genetic material and what is relationship with RNA. The DNA is a long polymer of deoxyribonucleotides, and the length of DNA is usually defined as number of nucleotides or a pair of nucleotides, referred to as the base pairs present in it. This also is the characteristic of an organism. For example, of the bacteriophage, known as into 174, has 5386 nucleotides. Bacteriophage lambda has 48502 base pairs. BP, Escherichia coli, has 4.6 into 10 s to 6 power 6 bp. And haploid content of human DNA is 3.3 into 10.9 bp. Let us discuss the structure of such a long polymer. Structure of polynucleotide chain. Let us replicate and the chemical structure of a polynucleotide chain, DNA and RNA. A nucleotide has three components as nitrogenous base, pentose, sugar ribose in case of RNA, and deoxyribose for DNA, and phosphate group. There are two types of nitrogenous bases: purines, adenine and guanine, and pyrimidines, cytosine, uracil and thymine. Cytosine is common for both DNA and RNA, and thymine is present in DNA. Uracil is uracil is present in RNA at the place of thymine. Nitrogenous base is to the OH and 1 degree Celsius pentose 1 carbon pentose sugar through N glycosidic linkage to form a nucleoside such as adenosine and adenosine, guanosine, and deoxyguanosine, the cytidine and deoxycytidine, the uridine and deoxyuridine and deoxyuridine and deoxythymidine. The when of phosphate group is linked to OH and 5 the carbon of or deoxynucleotide depending on upon the type of sugar present is formed. Two nucleotides are linked through 3,5 phosphodiester linkage to form a nucleotide. More nucleotides can be joined in such a manner to form a polynucleotide chain. A polymer thus formed has at one end a free phosphate moiety at a 5 prime end of sugar, which is referred to as 5 prime end of polynucleotide chain. Similarly, at the other end of the polymer, the sugar has a free OH and 3 prime carbon groups, which is referred to as 3 prime end of the polynucleotide chain. The base backbone of the polynucleotide chain is formed due to sugar and phosphates. The nitrogenous bases linked to sugar moiety project from the backbone. Figure 5.1. In RNA, every nucleotide residue has an additional OH group present at 2 position in the ribose. Also in RNA, the uracil is found at the place of thymine. 5-methyl uracil, another chemical name for thymine. DNA as an acidic substance present in nucleus was first identified by Friedrich Miescher in 1869. He named it as nuclein. However, due to technical limitation in isolating such a long polymer intact, the elucidation of structure of DNA remained elusive for a very long period of time. It was only in 1953 that James Watson and Francis Crick, based on the X-ray diffraction data produced by Maurice Wilkins and Rosalind Franklin, proposed a very simple but famous double helix model for the structure of DNA. One of the hallmarks of their proposition was the base pairing between the two strands of polynucleotide chains. However, this proposition was also based on the observation of Erwin Chargaff on that for a double stranded DNA, the ratios between adenine and thymine and guanine and cytosine are constant and equals 1. The base pairing confers a very unique property to the polynucleotide chains. They are said to be complementary to each other, and therefore if strand can be predicted. Also, if each strand from a DNA, let us call it as a parental DNA, acts as a template for synthesis of DNA strand, then the two double stranded DNA, let us call them as daughter or DNA, thus now produced would be identical to the parental DNA molecule. Because of this, the genetic implications of the structure of DNA become very clear. The slant features of the double helix structure of DNA are as follows. First, it is made of two polynucleotide chains where the backbone is constituted by sugar phosphate and the bases project inside. The second, the two chains have anti-parallel polarity. It means if one channel has the polarity 5 prime and 2 3 prime, and other has 3 prime to 5 prime. Third, the bases in two strands are paired through hydrogen bond IH bonds forming base pairs. Adenine forms two hydrogen Similarly, guanine is bonded with cytosine with three hydrogen bonds. Now, it generates approximately uniform distance between the two strands of the helix. Figure 5.2. The two chains are coiled in a right-handed fashion. The pitch of the helix is 3.4 nanometer, and a nanometer is 1 billionth of a meter. Then that is 10 is to power minus 3 meter, and there are roughly 10 bp in each turn. Consequently, the distance between a BP in a helix is approximately 0.34 nanometer. The plane of one base pair stacks over the other in double helix. This, in addition to hydrogen bonds, confers stability of the helical structure. Figure 5.3 compare the structure of purines and pyrimidines. Can you find out why the distance between two polynucleotide chains in DNA remains almost constant? The proposition of a double helix structure for DNA and its simplicity in the explaining the genetic implication become revolutionary very soon. Francis Crick proposed the central dogma in molecular biology, which states that the genetic information flows from DNA to RNA, RNA to protein. In some viruses, the flow of information is in very direction, that is from RNA to DNA. Can you suggest the simple name to the process? Packing of DNA helix taken a distance between two consecutive base pairs, also 0.34 millimeter, 0.4 into 10 is to minus 9 meter. If the length of DNA double helix in a typical mammalian cell is calculated simply by multiplying the total number of BP with distance between two consecutive BP, that is 6.6 into 10 is to 9 BP and 2.34 into 10 is to 9 meter per BP, it comes out to be approximately 2.2 meters length. That is far greater than the dimension of a typical nucleus, approximately 10 is to minus 6 meter. How is such a long polymer packaged in a cell? If the length of Escherichia coli DNA is 1.36 millimeter, can you calculate the number of base pairs in Escherichia coli? Now, the prokaryotes, such as Escherichia coli, though they do not have defined nucleus, the DNA is not scattered throughout the cell. The DNA, being negatively charged, is held with some proteins that have positive charges in a region termed as nucleoid. The DNA is nucleoid or is organized in large loops held by proteins. In eukaryotes, this organization is much more complex. There is a set of positively charged basis proteins called a histone. A protein acquires charge depending upon the abundance of amino acids. The residues with charged side chains, histones, are rich in the basic amino acids. Residues, lysine and arginine, both the amino acid residues carry positive charges in their side chains. Histones are organized to form a unit of 8 molecules called histones octamer. The negatively charged DNA is wrapped around the positively charged histone positively charged positively charged typical positively charged histone octamer to form the structure called a nucleosomes. Figure 5.4a. A typical nucleosome contains 200 BP base pair of DNA helix. Nucleosomes constitute like stained colored bodies seen in nucleus. The nucleus is lets nucleotides is chromatin are seen as beads on string structure when viewers under the electron microscope. EM, the figure 5.4b. Theoretically, how many such beads nucleosomes do you imagine are present in a mammalian cell? The beads on string structure in chromatin is packaged to form chromatin fibers that are further coiled and condensed at metaphase stage of cell division to form chromosomes. The packaging of chromatin at higher level requires additional set of proteins that collectively are referred to as non-histone chromosomal NHC proteins. In a typical nucleus, some new region of chromatin are loosely packed and stains light and are referred to as euchromatin. The chromatin that is more densely packed and stained dark are called heterochromatin. Euchromatin is said to be transcriptionally active chromatin, whereas heterochromatin is inactive. Rajan! 5.2, the search for genetic material. Even through the discovery of nucleon, the measure and the proposition of for principles of inheritance by Mendel were almost at the same time, but that the DNA acts as a genetic material took long to be discovered and proven. By 1926, the course to determine the mechanism for genetic inheritance had reached the molecular level. Previous discoveries by Gregor Mendel, Walter Sutton, Thomas Hunt Morgan, and numerous other scientists had narrowed the search for to the chromosomes located in the nucleus of most cells. But the question of what molecule was actually the genetic material had not been answered. Transforming principles in 1928, Frederick Griffith, in a series of experiments with Streptococcus pneumonia, bacterium responsible for pneumonia, witnessed miraculous transformation in the bacteria during the course of his experiment. A living organism, bacteria had changed in physical form when Streptococcus pneumococcus bacteria are grown on a culture plate. Some produce smooth, shiny colonies, yes, while others produce rough colonies. Are no, this is because the S strain bacteria have a mucus polysaccharide coat, while R strain does not. Mice infected with the S strain virulent die from pneumonia infection, but mice infected with the R strain do to develop pneumonia. S strain inject into mice die. R10 inject into mice live. Grint was able to kill bacteria by hitting them. Now he observed that heat killed yes strain bacteria injected into mice did not kill them when the yes strain heat killed injected into mice life yes strain heat killed plus yes strain life inject into mice die. Injected a mixture injected a mixture of heat killed yes and live wire bacteria. The mice died. Moreover, he recovered living yes bacteria from dead mice. He concluded that our strain bacteria had somehow been transformed by the heat killed yes strain bacteria. Some transforming principle transferred from the heat killed S strain had enabled the SR strain to synthesis the smooth polysaccharide coat and become virulent. This must be due to the transfer of genetic material. However, the biochemical nature of genetic material was not defined from his experiment. Biochemical class characterization of transforming principle prior to the work of Oswald Avery, Colin MacLeod, and MacLeod McCarthy, 1933 to 1944. The genetic material was through to be protein. They worked to determine the biochemical nature of transforming principle of Griffith experiment. Then they purified biochemicals, proteins, DNA, RNA, etc. from the heat killed yes cells to see which ones could transform live far cells into yes cells. They discovered that the DNA alone from yes bacteria caused R bacteria to become transformed. They also discovered that protein digesting enzymes, proteases, and the RNA digesting enzymes, RNases, it did not affect transformation, so as transforming substance was not protein or RNA digestion with DNA's transformation. They concluded that DNA is the hereditary material, but not all biologists were convinced. Can you think of any difference between DNAs and DNA and DNAs? The genetic material is DNA. The unequivocal proof that DNA is the genetic material came from the experiment of Alfred Hershey and Martha Chase. 1952, they worked with viruses that infect bacteria called the bacteriophage. The bacteriophage attached to the bacteria and its genetic material then enters the bacterial cell. The bacterial cell threads the viral genetic material as if it was its own and subsequently manufactures more virus particles. Hershey and Chase worked to discover whether it was protein or DNA from the viruses that entered the bacteria. They grew some viruses on the medium that contained radioactive phosphorus and some others on medium that contained radioactive sulfur. Viruses grown in the presence of radioactive phosphorus contained phosphorus, but the protein does not. Similarly, viruses grown on radioactive sulfur contain radioactive protein but not radioactive DNA, and because DNA does not contain sulfur, the radioactive phases were allowed to attach to Escherichia coli bacteria. Then, and the infection proceeds, the viral codes were removed from the bacteria by agitating them in a blender. The virus particles were separated from the bacteria by spinning them in a centrifuge. Bacteria which was infected with viruses that had a radioactive DNA were radioactive indicating that DNA was the material that passed from the virus to the bacteria. Bacteria that were infected with viruses that had radioactive proteins were not radioactive. This indicates that proteins did not enter the bacteria from the viruses. DNA is therefore the genetic material that is passed from virus to bacteria. Figure 5.5. Properties of genetic material, DNA, viruses, and RNA. From the foregoing discussion, it is clear that the debate between proteins versus DNA as the genetic material was unequivocally resolved from Hershey or Chase experiment. It became an established fact that it is DNA that acts as genetic material. However, it subsequently became clear that In some viruses, RNA is the genetic material. For example, tobacco mosaic viruses, QB, bacteriophage, etc. Answer to some of the questions, such as why the DNA is the predominant genetic material, whereas RNA performs dynamic functions of messenger and adapter, has to be found from the differences between the chemical structures of the two nucleic acid molecules. Can you recall the two chemical differences between DNA and RNA? A molecule that can act a genetic material must fulfill the following criteria. First, it should be able to generate its replication. Second, it should be stable chemically and structurally. Third, it should be provide the scope for slow changes, mutation that are required for evolution. Fourth, it should be able to express itself in the form of Mendelian characters. If one examines such each requirement one by one, because of rule of base pairing and complex complementarity, both the nucleic acid, DNA and RNA have the ability to direct their the other molecule in the living system, such as proteins, for fail to fulfill first criteria itself. The genetic material should be stable enough not to change with different stages of life cycle, age, or with change in physiology of the organism. Stability as one of the properties of genetic material was very evident in Griffith's with transforming principle itself, that hit which killed the bacteria, at least did not destroy some of the properties of genetic material. This now can easily be explained in light of the DNA that the material. This now can easily explained in light of the DNA that the two strands, being complementary, if separated by heating, so come together when appropriate condition are provided. Further, to prime OH group present at every nucleotide in RNA is a reactive group and makes RNA labile and easily degradable. RNA is also now no to the catalytic, hence reactive. Therefore, DNA chemically is less reactive and structurally more stable when compared to RNA. Therefore, among the two nucleic acids, and the DNA is a better genetic material. In fact, the presence of thymine at the place of uracil also confers additional stability or to DNA. A detailed the discussion about this requires understanding of the process of repair of the DNA, and you will study in this processes in higher classes. Both DNA and RNA are able to mutate. In fact, RNA being unstable mutate at a faster rate. Consequently, viruses having RNA genome and having shorter lifespan mutate and evolve faster. RNA can directly code for the synthesis of proteins, hence can easily express the characters. DNA, however, is dependent on RNA for synthesis of proteins. The protein synthesizing machinery has evolved around RNA. The above discussion indicates that both RNA and DNA can function as genetic material, but DNA, being more stable, is preferred for storage of genetic information. For the transmission of genetic information, RNA is better. RNA world. From foregoing discussion, an immediate question becomes evident, which is the genetic material. It shall be discussed in detail in the chapter on chemical evolution, but briefly we shall highlight some of the facts on and points. RNA was the first genetic material. There is now enough evidence to suggest that essential life processes such as metabolism, translation, splicing, etc. evolved around RNA. RNA used to act as genetic material as well as a catalyst. There are some important biochemical reactions in living systems that are catalyzed by RNA catalysts and not by protein enzymes. But RNA being a catalyst was reactive and hence unstable. Therefore, DNA has evolved from RNA with chemical modifications that make it more stable. DNA being double stranded and having a complementary strand further resists changes by evolving a process of repair. Replication 5.4.